an associate professor at the University of the Philippines, Cebu. She teaches general biology courses and advanced courses in plant biology since 2003. She also mentors both undergraduate and postgraduate students in plant-related thesis research works. Mom Cadiz obtained her PhD in Science from the University of Melbourne, Australia, Master of Science in Botany from UP Los Baños, and Bachelor of Science in Biology from UP Cebu. Her areas of interest include plant ecophysiology, vegetation dynamics, forest fire ecology, plant invasion ecology, and invasive plants, and also plant diversity. Our speaker this afternoon already presented various research in different conferences and published several articles. Our speaker this afternoon, Dr. Geofe Cadiz.
Good afternoon, everyone. First, I would like to extend my gratitude to the Conserve Kaigangan team, headed by Dr. Jun Buot, for inviting me to share my work with you. Sir Jun was my thesis advisor back then when I took my master's degree in botany in UP Los Baños. And what I will be presenting is actually part of my master's thesis. This was completed many years back. And unfortunately, since then, I did not have the chance of working in forest over limestone anymore. And I was working on a different type of forest, which is a fire-prone temperate forest in Australia, dominated by the flammable eucalyptus species. I was particularly working on the forest dominated by the second tallest tree in the world, the Eucalyptus regnans. Now, Australia is one of the mega diverse countries, just like our country, the Philippines. But unlike Australia, the Philippines is one of the hottest hotspots worldwide, as identified by Myers and colleague in 2000. When we say biodiversity hotspots, these are areas with exceptional concentrations of endemic species and experiencing exceptional loss of habitat. Our country is particularly rich in biodiversity, but at the same time, having the lowest percentage of remaining primary vegetation cover among all other countries. Now, these facts highlighted the importance of documenting our biodiversity and putting in place measures and strategies for conservation and protection. Now, key biodiversity areas in the Philippines were identified to target conservation action on, and 6 of the 228 identified KBAs are in Cebu Island. The KBAs were identified based on the conservation priority areas set forth by the Philippine Biodiversity Conservation Priority Setting Program. 7 of these CPAs are found in Cebu Island alone, and were all given an extremely high critical priority level. Now, in one of these 7 CPAs, the Tabunan Forest was where I conducted my study on the vegetation characteristic of an extremely high critical forest over limestone. Tabunan Forest is an old growth forest located in central Cebu and included in the Central Cebu Protected Landscape, which is one of the key biodiversity areas in Cebu Island. Tabunan Forest is part of the remaining 0.3% old growth forest fragment in Cebu. And this forest has an elongated shape. It is just a small patch with an elevation ranging from 500 to 880 meters above sea level. This forest has received global attention because it is where the endemic bird Cebu flower pecker was rediscovered. And this bird, scientifically known as Dickeum quadricolor, was thought to be extinct in 1906 until its rediscovery in 1992 in Tabunan Forest. The Cebu flower pecker is a critically endangered bird, commonly found in the closed forest canopy and near flowering or fruiting plants. Tabunan Forest is also a home to other endemic and threatened bird species in Cebu. Also, the critically endangered Philippine tube-nosed fruit bat, the critically endangered endemic plant, the Cyanometra cebuensis, and the endangered endemic plant, Cinnamomum cebuense. But despite the fact that Tabunan Forest is in a strict protection zone, there is that continuous threat of exploitation by the local people and the threat from edge effect owing to its very, very small size. Now, I studied the vegetation characteristic of this forest in view of its value, the richness of its biodiversity, and the threat that it is facing. And so the objectives or the study aimed to characterize the vegetation composition and diversity in Northeast Tabunan Forest through vegetation survey and to identify environmental factors associated with the plant diversity in Tabunan Forest through multiple regression analysis. And the vegetation survey was conducted in 10 20 by 20 meter plots for woody vegetation and in 2 by 5 meter plot within the bigger plot for herbaceous ground vegetation. And 288 taxa were surveyed, representing 135 genera and 68 families. Most of these taxa were angiosperms. 225 were identified up to the species level in 61 families. Only 2 gymnosperms in 2 families were found in the survey. These were Netumnemon and Podocarpus species. And then there were also 6 pteridophytes or fern species belonging to 5 families. These are Cristella species, Lygodium circinatum, Nephrolepis cordifolia, Pernifrium species, Pteris armata, and Tectaria dissecta. I would like also to emphasize that 3 dipterocarps were also part of the woody angiosperm in Tabunan Forest. One is Hopea philippinensis, which is a Philippine endemic and an endangered species. Another is Paraschorea malaanunan, and Shorea contorta or lawaan, which is also a Philippine endemic and categorized as vulnerable species. Now, in the Philippines, dipterocarps are almost always part of the floristic composition at lower altitude. The most represented families were Moraceae family with 24 species. Then we have Meliaceae family with 22 species and Araceae or the aroid family with 18 species. And the most represented genera were Ficus, which belongs to the most represented Moraceae family. Then we have Aglaea, which belongs to the Meliaceae family, and Garcinia under the Clusiaceae family. These are pantropical families and genera and are also common in other forests over limestone. Now, in terms of DBH, majority of the trees found in each quadrat were in the smaller diameter classes. This is the general trend in tropical forests because there are more trees in the lower strata, including many young individuals of species that may reach the canopy when they mature, as well as species that will not do so. So the highest proportion of trees in the study area had a DBH of less than 11 cm. Furthermore, in almost all of the quadrats, trees with DBH of more than 30 cm had the lowest proportion. This DBH class of trees contributed only less than 10% of the total trees per quadrat. The highest DBH of 159.2 centimeters was recorded from the species, the Dipterocarp Shorea contorta. Now, in the southwestern portion of Tabunan Forest, the highest DBH sampled in the study of Orlanes in 2002 was just 97 centimeters. Now, in terms of height, majority of the trees in almost all of the plots were 6 to 10 meters tall. Trees with height of more than 20 meters in the northeastern portion of Tabunan were present in low proportion. Few emergence were observed with heights ranging from 40 to 65 meters. And these include the Dipterocarp Shorea contorta with a height of 50 meters and also Planchonelia species with a height of 55 meters. Now, the plant species composition in Tabunan Forest is highly diverse, with a Shannon index value of 4.51. One factor that contributes to the high diversity of species in Tabunan Forest is the low taxa frequency. Almost half of the families and 75% of the genera were only represented by single individuals. This high diversity is also a characteristic of tropical rainforests where most plant species, especially the trees, are characterized by very small numbers of individuals. Now, I investigated what environmental factors were associated with the variations in diversity of the 10 sampling plots through a stepwise multiple regression analysis between the diversity index and environmental factors. These environmental factors include temperature. We have topographic factors like elevation, slope grade, microtopography, and also soil factors like soil pH, the soil cation exchange capacity or CEC, the nitrogen, phosphorus, potassium content of the soil, the texture of the soil. Then we have the organic matter content of the soil, and the water holding capacity of the soil. The soil factors were measured from soil samples collected in all sampling plots. Now, of all these factors, only the water holding capacity was significantly associated with the variation of species diversity of the 10 sampling plots. And this factor explained 49% of the total variation. Now, the water holding capacity is dependent upon the organic matter in the soil and soil particle size. Soil type and quality can change a great deal, even over short distances, thus promoting a variable mosaic of natural vegetation. However, in other Philippine forests, such as in Mount Akiki and Mount Pulag, as studied by Sir Jun Buot and colleague, altitude is the main causal factor influencing the diversity pattern. Species diversity in those mountains generally increased with altitude. Now, I did some more assessment into the species composition of the northeastern portion of Tabunan and determine whether the species are endemic or indigenous or introduced, and whether they are commonly found in primary or disturbed secondary forest, based on Merrill's enumeration of Philippine flowering plants and CAUS digital flora of the Philippines. And I also noted the conservation status of the species. The table being shown here is the output table. This is actually a long table. I'm just showing a portion. And based on this assessment, all the species in Tabunan Forest are native to the Philippines. There are no introduced or exotic species. In addition, of all those species, 28% are endemic. That is, they are only found in the Philippines. Also, there are 4 threatened species. One is categorized as critically endangered. This is the Dipterocarp hopea philippinensis. And 3 are in the vulnerable category. We have Juspirus blancoi, the endemic Mangifera altissima, and another endemic Shorea contorta, the dipterocarp. Now, the presence of endemic and threatened species highlight the necessity of putting a premium on the protection of tabunan forest because it holds many species which are only found in the Philippines and nowhere else in the world. Lastly, the assessment also shows that 26% of the identified species with known distributions are species commonly found in secondary forests. Secondary forests are forests that have been disturbed in some way, naturally or unnaturally. And the presence of these species in tabunan indicates that this area has undergone considerable disturbance. And this is consistent with the observations of anthropogenic disturbances in tabunan forest. And here I listed those. Human trails are obvious, no, inside the forest. Established foot trails lead to the viewing deck for bird watchers. They also have less obvious trails leading further into the forest interior. Um, Local residents and tourists usually go inside the forest. The former are either part of the forest protection programs because they have this wardening program. They have tanod in the forest. Or those people who cut trees and harvest rattan, gather food, harvest birds' nest, and also hunt monkeys and birds. Now, the tourists are usually foreigners who visit there, mostly to see the Cebu flower pecker, the famous Cebu flower pecker, and the Cebu black shama, and the other birds. No, researchers also occasionally visit Tabunan Forest for study. Now, in closing, a number of protection and conservation programs and initiatives had been implemented for Tabunan Forest by different groups, such as the Tabunan Forest Biodiversity Conservation Project by the Darwin Initiative, um, the Philippines Wetland and Wildlife Conservation Foundation also did some project protection conservation programs and initiatives there. The Flora and Fauna International, and then also a foundation was created, the Cebu Biodiversity Conservation Foundation, and we also have other entities like, of course, the DENR, the local NGO, and farmers cooperative. So these groups recognize the fragility of this small forest and its crucial importance for biodiversity conservation, being one of the last remnants of Cebu's natural heritage. Now, before I end, I would like to acknowledge the following for their support and valuable input to this project. For UP Visayas for the funding, to Sir June, Ma'am Rachel, Sir Nestor, and also Professor Archie and Dr. Araneta for their valuable inputs to this study, to the late Mr. Leonard Ko for identifying the species, to Mr. Silor and Mr. Babiera from the Central Cebu Protected Landscape for allowing me to conduct my study in Tabunan Forest during those years. And my thesis workforce, Tatay King, Kyle, Mark, and Geraldine, and the Son family for the warm accommodation during my fieldwork. Thank you very much.